Good morning, everyone. Today's topic is physiology of neuromuscular junction, that is, the junction between nerve and muscle. So, how the signal transmits from the nerve to the muscle, that is dealt with in this topic. Okay, so we will see the contents like definition, structure, acetylcholine synthesis, storage and release, and sequence of events which take place at the neuromuscular junction, the clinical importance of the neuromuscular junction and its applied aspects. So this is also a very important essay question for you. So pay attention. Coming to the definition of the neuromuscular junction, it is the synapse or junction of axon terminal of a motor neuron with motor end plate, and it is responsible for initiation of action potentials across the muscle surface, ultimately causing the muscle to contract. So once again, it is a junction or synapse of the axon terminal of a motor neuron with motor end plate. Motor end plate means it's present on the muscle. So muscle thickening at one point is the motor end plate, responsible for initiation of action potentials across the muscle surface, ultimately causing the muscle to contract. Or simply you can just write as the junction between the motor nerve and skeletal muscle fiber. So the parts of the neuromuscular junction are presynaptic membrane, synaptic cleft and postsynaptic membrane. So this is when we see the structure under the electron microscope. Okay, so this is one picture, and then so this is the detailed picture of the neuromuscular junction. So underneath the nerve ending, muscle membrane of the motor end plate. So this is the motor end plate. You can, as I have told you, the surface of the muscle where the nerve ends. Okay, underneath the nerve ending, the muscle membrane of motor end plate is thrown into folds. Okay, like this, folds. These are called palisades. These folds increase the surface area on which the neuromuscular transmitter can act. So the neurotransmitter released from the presynaptic membrane will act on these motor end plates. So the surface area is increased by these foldings called palisades. So at the terminal nerve endings, there are many mitochondria and minute vesicles or synaptic vesicles or granules. Okay, all these are the synaptic vesicles or granules. So which contains small packets of chemical transmitter called acetylcholine, responsible for synaptic transmission. So nerve membrane, which is in close approximation with the muscle membrane, is known as the presynaptic membrane, while the muscle membrane is known as the postsynaptic. So this membrane is the presynaptic membrane and this membrane is the postsynaptic membrane. And the space between the two membranes is called synaptic cleft. So this minute space is there, no, that is synaptic cleft, which is 50 to 100 nanometers wide, and it is filled with ECF. So the nicotinic ACH receptors are found in the postsynaptic membrane at the junctional folds. So these are so called because they are stimulated by both nicotine and ACH and inhibited by curare. Okay, so this is the brief structure of the neuromuscular junction. This is again one more picture. So this is the presynaptic membrane terminal containing many mitochondria and vesicles or granules. So and the postsynaptic membrane is thrown into folds, palisades, which increases surface area, and the receptors are present on the postsynaptic membrane. So this is one more picture. You can see the nerve terminal ending on the muscle. So this is a motor unit. So motor unit is each motor neuron from the ventral horn runs uninterrupted up to the muscle, splits into a functional group called the motor unit. Single neuron supplying a group of muscle fibers, and this motor unit contract and relax as a unit. So it consists of one motor neuron and all the muscle fibers it stimulates. Okay, so you can see from the spinal cord one motor neuron is coming and it innervating all the fibers in a muscle. So this figure shows three motor units, one purple colored, one blue colored and one red colored one. So each nerve will innervate a group of muscle. In a muscle there are muscle fibers. Okay, right? So the nerve fiber with its branches and supplying the muscle fibers is called a motor unit. And the branching nerve terminals invaginate into the surface of muscle fiber but lie outside the sarcolemma. Hence, the synaptic cleft is formed. Usually around 20 to 30 nanometers or 50 to 100. It's given in different books, and ACH receptors are present on the postsynaptic or postjunctional membrane. So this is again one more picture. So this is the vesicle, synaptic vesicle containing ACH neurotransmitter or the granules. Okay, so this ACH synaptic vesicle is surrounded with this protein, synaptophysin, synaptobrevin, synaptomin, synapsin. Okay, like this various proteins are present around this vesicle. And there are calcium channels on the presynaptic nerve terminal, and also sodium channels and potassium channels, and choline will easily enter into the presynaptic nerve. So how the ACH is formed? By means of acetylcholine, CoA and choline when combined with acetyl transferase, it will form acetylcholine. Okay, so we will see the synthesis, storage and release of the acetyl. So this is the picture showing the synthesis and storage and release of the acetylcholine. So it is synthesized in the mitochondria with a union of choline and acetyl-CoA. In addition to these two, it requires ATP and glucose. Okay, with the exception of choline, all components required for the synthesis are present within the mitochondria and gain ready access to them. So once formed, it is temporarily stored in the vesicles. Okay, and after that it is released whenever there is nerve transmission at the neuromuscular junction, and this vesicle contains quantum or packets of few hundreds or thousand molecules of ACH. So nerve impulse causes fusion of vesicles with the membrane, presynaptic membrane, by increasing the permeability of calcium. So calcium helps in fusion by changing ionic composition and thereby decreasing the flow of exoplasm. Therefore, vesicles easily come in contact with the membrane. The amount of transmitter released is directly proportional to the calcium. So, this ACH release is directly related to the calcium in the ECF. So, it helps in fusion of vesicles with the membrane, and vesicles easily come in contact with the membrane. So for the release of the ACH, calcium channel, calcium influx is necessary. Okay. So we will see briefly about the ACH receptors. They are synthesized inside the muscle fiber. It is composed of 5 subunits, and on the basis of these subunits, they are classified as adult, mature or junctional, fetal or extrajunctional. So today we will see only adult or mature acetylcholine receptors. They are present in the postsynaptic membrane of motor end plate and are of nicotinic types. These exist in pairs. It consists of protein made up of 1000 amino acids and made up of 5 protein subunits designated as alpha, beta, delta and epsilon. So alpha has alpha 1 and beta has beta 1 part. So joined to form a channel that penetrates through and projects on each side of the membrane. Each receptor has a central funnel-shaped core which is an ion channel 4 nanometer in diameter at entrance, narrowing to less than 0.7 nanometer within the membrane. The receptor is 11 nanometer in length and extends 2 nanometer into the cytoplasm of the muscle cell. So don't worry about all these details. So this is just for your knowledge. The receptor has two gates, an upper voltage dependent and lower time dependent gates. So when acetylcholine receptors bind to pentameric complex, they induce a conformational change in the proteins of alpha subunits which open the channel, and it occurs only if it binds to both alpha binding sites. Okay, so when the receptors bind to pentameric complex, that is they form a 5 pentameric means 5 subunits. They induce a conformational change in the proteins of the alpha subunits which opens the channel, and this opening occurs only if it binds to both alpha binding sites. So for ions to pass through the channel, both the gates should open. So for ions to pass through, both the gates should open. Cations flow through the open channel, sodium and calcium in and potassium out, thus generating end plate potential. So calcium and sodium move in and potassium go out, generating the end plate potential. Sodium ions are attached to inside of the cell, which induces depolarization. Sodium, after entering into the cell, it attaches to the inside of the cell. So sodium influx causes depolarization. So these are the low conductance immature channels, and these are the high conductance mature channels. So mature and immature are varying, differ in the conductance, that is the ionic conduction, movement of ions through the channels. So immature receptors or channels have increased sensitivity to depolarizing agents, decreased sensitivity to non-depolarizing agents. So depolarizing means sodium, when it enters inwards the muscle, it causes depolarization. Sodium influx is responsible for depolarization, that we will see in the coming classes, nerve action potential, and it stays open for a longer time. Hence, increased efflux of intracellular potassium occurs. Altogether, it can cause lethal hyperkalemia. So now this is a picture showing the sequence of events taking place at the neuromuscular junction during the nerve impulse transmission. So when the nerve action potential reaches the presynaptic nerve ending, it causes sodium influx. So the ACH synaptic vesicles reach the membrane, presynaptic membrane, and it fuses with the membrane and causes exocytosis. So ACH, sodium influx, causes depolarization, and that leads to opening of calcium channels. Calcium channels are of two types, P and L channels. So calcium entry into the nerve occurs, and that leads to ACH release. So ACH, after its release, diffuses across a short distance to the postsynaptic membrane or motor end plate. So these receptors, it binds to the post-junctional receptors. Again, sodium channels on the postsynaptic membrane, they open, and that causes depolarization, which leads to the action potential generation on the postsynaptic membrane. So this transmission of action potential allows sarcolemma to open tubular calcium channels. So on the muscle, there are again tubular calcium channels present. So that will lead to finally muscle contraction. So this is the final response of the neuromuscular transmission. The nerve signal will lead to final response of muscle contraction. Okay, so these are the events which take place starting from the nerve, presynaptic membrane, synaptic cleft, and then postsynaptic membrane, finally muscle contraction. So this is in sentence formation. When a nerve impulse reaches the terminal, it opens calcium channels. Calcium diffuses from the ECF into the axon terminal. Okay, so this is here, calcium diffuses from ECF into the axon terminal. This calcium releases ACH from the vesicles by process of exocytosis. So one nerve impulse can release 125 ACH vesicles like this. In the presynaptic nerve terminal, 125 vesicles it can release. only one nerve impulse. The quantity of ACH released by one nerve impulse is more than enough to produce one end plate potential. ACH combines with its receptors in the subneural clefts. This opens sodium channels and sodium diffuses into the muscle, causing local non-propagated potential called end plate potential. So this end plate potential is very important in producing muscle contraction, whose value is 50 to 75 millivolts. This EPP triggers a muscle action potential which spreads down inside the muscle to make it contract. So after the signal has been transmitted, what happens to the remaining vesicles? Discharged vesicles are rapidly replaced from the reserve stores. So they are replaced with the new vesicles. Reserve vesicles anchored to cytoskeletal actin by synapsin. So in the previous slide I have shown you these vesicles have many proteins, anchoring proteins like synapsin, synaptobremin, synaptognomin. Okay, so they reserved vesicles are anchored, already attached to the cytoskeletal actin by synapsin. So calcium entry during the initial discharge process binds to calmodulin. So stimulates protein kinase 2 which phosphorylates synapsin, and thus reserved vesicles are freed. So when the calcium enters into the presynaptic neuron, it binds to calmodulin which further will stimulate protein kinase 2 phosphorylating the synapsin. That means that attached chains are being broken down by the calcium entry, thus freeing the reserve vesicles. And docking of the vesicle, that is attaching of vesicle to the presynaptic membrane. Docking means attaching, and subsequent discharge of acetylcholine by exocytosis involves several other proteins. Membrane protein called SNARE, that is soluble N ethylmatrimide sensitive attachment proteins, are involved in fusion, docking and release of acetylcholine at the active zone. So other than the calcium influx, there are membrane proteins called SNAREs which help in fusion of the vesicle, docking of the vesicle and release of ACH. So this SNARE proteins include synaptic vesicle protein synaptobremin, syntaxin and SNAP25. So like this, the docking and fusion and release of ACH from the vesicle occurs. Okay, neurotransmitter uptake, translocation, docking is attaching, priming and fusion or exocytosis, and then again endocytosis, and then translocation, endosome fusion, and this goes on continuous as a cycle. The released acetylcholine diffuses to the muscle type nicotinic acetylcholine receptors which are concentrated at the tops of junctional folds of membrane of the motor end plate. So receptors are concentrated at the tops of the junctional folds of the postsynaptic membrane. Binding of ACH to these receptors increases sodium and potassium conductance of the membrane, and resultant influx of sodium produces depolarizing potential, that is end plate potential. Okay, so when the ACH binds to these receptors, that increases the sodium and potassium conductance into the membrane, that causes depolarization or end plate potential. The current created by the local potential depolarizes the adjacent muscle membrane to the firing level. That is, firing level means threshold level. So again the same which I have told you in the flow chart. Increases sodium potassium conductance, depolarization of end plate potential production, and after the production of end plate potential, it depolarizes the adjacent sarcolemma to firing level. That's how continuously the entire sarcolemma or the entire muscle fiber length is being depolarized, producing the uh, muscle contraction. Acetylcholine is then removed by acetylcholine esterase from the synaptic cleft, which is present in high concentration at the neuromuscular junction. Action potential generated on either side of the end plate are conducted away from the end plate in both the directions along the muscle fiber. So from the point, it is conducted in both the directions so that entire muscle fiber length is being stimulated. The muscle action potential in turn initiates muscle contraction. So we'll briefly see about the sodium channels. We are always talking about the sodium influx, right? So this sodium channel is cylindrical in shape. It two ends has uh, acts at the ends only act as a gates, and both should be open to allow passage of ions. Voltage dependent gate is on one side and time dependent gate is on other side. So voltage gate is closed in resting state and opens only on application of depolarizing voltage, and it remains open or as long as the voltage persists. And the time dependent gate is normally open at rest, closing a few milliseconds after the voltage gate opens and remains closed as long as the voltage gate is open. It reopens after the voltage gate closes. So when one is open, other closes. Only when both are open, there is influx of sodium occurs. So the channel is patent, allowing sodium ions only when the gates are open. So resting state, how the channel will be: voltage gate closed, time gate open, channel closed, finally channel closed. Depolarization, voltage gate, time gate open, channel open, and within a few milliseconds voltage gate open, time gate closed, so channel closed. End of depolarization, both voltage gate closed, time gate will be open. So resting state, depolarization, end of depolarization, time gate will be open, and the voltage gate is closed in resting and end of depolarization. So channel is open only in the depolarization state, that is when both the gates are open. So this is little bit confusing, but try to understand it. See the previous slides and then you try to understand it. And the calcium role is: concentration of calcium and length of time during which it flows into the nerve ending determines the number of quanta, that is packets of ACH in the synaptic vesicles. Calcium current is normally stopped by the outflow of potassium. Calcium channels are specialized proteins which are opened by voltage change accompanying action potentials. Part of calcium is captured by proteins in the endoplasmic reticulum and is sequestrated. Remaining part is removed out of the nerve by sodium-calcium antipode system. The sodium is eventually removed from the cell by ATPase. So this is removal of calcium, whatever that has come inside the nerve terminal. That is how it is replaced or taken back by these three ways. One is again it is taken back into the endoplasmic reticulum, and one more is sodium calcium antipode system, and one more is ATPase. The sodium is removed from the cell by ATPase. And coming to the most important, acetylcholine esterase enzyme. So without this enzyme, if we imagine, if the enzyme acetylcholine esterase is not present, there will be continuous presence of or release of ACH with one nerve impulse, and there will be continuous contraction of the muscle taking place. So acetylcholine has to be taken back or disassociated by the acetylcholine esterase enzyme for the required muscle contraction to take place and for the muscle to relax. And this protein enzyme is secreted from the muscle but remain attached to it by thin stalks of collagen attached to the basement membrane. Acetylcholine molecules that don't interact with the receptors are released from the binding site and are destroyed almost immediately by acetylcholine esterase in less than 1 milliseconds. So imagine how fast it is after its release into the junctional cleft. So within less than 1 milliseconds, it is destroyed almost immediately. So different pools of acetylcholine in the nerve terminal. So one is active zone, small size, very close to the synaptic nerve terminal ready to release, and other is reserve storage, that is large size. These vesicles have been recycled after their use. When the nerve is repeatedly stimulated, these vesicles are mobilized from zone 2 to zone 1 and more ACH become available for impulse transmission. And till now we have seen the entire sequence or transmission of nerve muscle or the events in the neuromuscular junction, and now we will see the neuromuscular blockers, that is action of drugs on the neuromuscular so can we block this neuromuscular transmission? Yes, it can be blocked by two ways: inhibiting the release of ACH by example botulinum toxin, and inhibiting the action of ACH on its receptor by depolarizing muscle relaxant and non-depolarizing muscle relaxant. So first we will see how to inhibit the release of ACH. By botulinum toxin, it exerts its lethal effect by blocking the release of ACH. It cleaves needs proteins, preventing releasing the vesicles. These poisons most frequently result from improperly canned food contaminated with Clostridium bacteria. So action of ACH on the receptors is blocked by non-depolarizing and depolarizing muscle relaxants. So this non-depolarizing impair or block neurotransmission by competitively preventing the binding of acetylcholine to its receptor. These muscle relaxants bind with the same receptors and close the channel. So there is no chance of ACH binding to the receptor. Two molecules of ACH per receptor are needed to open the channels. Single molecule of NMDR, that is non-depolarizing muscle relaxant, for single receptor is adequate to prevent the opening of channel. So the final outcome is blockage or transmission of the signal, and it depends on the relative concentrations of chemicals and their comparative affinities for the receptor. So the non-depolarizing muscle relaxant action, how it can be reversed. So normally acetylcholine esterase enzyme, what it does, it destroys ACH and removes it from competition for a receptor. But here what is happening, already the receptors are being blocked, so more ACH is required. So if we block this or inhibit this acetylcholine esterase enzyme by help of drugs like neostigmine, what happens, finally acetylcholine concentration rises. So higher concentration shifts the competition between ACH and NDMR in favor of ACH, thereby improving the chance of two ACH molecules binding to a receptor. So thus leading to the nerve transmission at the neuromuscular junction. This causes reversal of NDMR effect. So the depolarizing muscle relaxant, how they act means they act same like ACH acetylcholine by binding same alpha subunit and initiate the ACH destroyed by the enzyme acetylcholine esterase in less than 1 milliseconds. So succinylcholine or suxamethonium or and one more example is decamethonium. So their duration of action is very short, 5 minutes. So these drugs act by persistent depolarization. Okay, so continuously depolarization is taking place. What happens, so energy exhaustion occurs, which no further generation of ATP is resulting in muscle relaxation. Okay, so these are very short acting drugs, so there is no need of antidote. So they cause elimination, succinylcholine elimination by hydrolysis by plasma or pseudocholine esterase. So they will be immediately destroyed. So there is no antidote after their action. Again ACH normally will come and do its action. So perijunctional area of end plate have two different types of membranes. One is end plate membrane and true muscle membrane. And next, coming to the applied aspects of the neuromuscular junction. So myasthenia gravis, I think most of you may be knowing this about this disease. So it is an autoimmune disorder characterized by fatigable weakness of skeletal muscles. And weakness results from an antibody-mediated immunological attack directed at the acetylcholine receptor or receptor-associated proteins in the postsynaptic membrane of neuromuscular. So what it is basically, it is an autoimmune disorder characterized by fatigable weakness of skeletal. Fatigable means by usage the muscle becomes fatigued and then they become weak. And weakness results from antibody-mediated immunological attack directed at the acetylcholine receptors present on the postsynaptic membrane. So patients with MG are sensitive to non-depolarizing neuromuscular blockers and are resistant to acetylcholine, that is a depolarizing neuromuscular blocker. So sensitive to non-depolarizing. So what are the non-depolarizing muscular blockers? That is tuberculin, galamine and curare. Okay, so non-depolarizing neuromuscular blockers. So one more applied aspect is Lambert-Eaton syndrome, otherwise myasthenic syndrome. It is caused by IgG antibodies to voltage-gated calcium channels. It leads to increased sensitivity to both SCH, succinylcholine, and non-depolarizing muscle relaxants. So it is associated with small cell carcinoma of lung, sarcoidosis, thyroiditis, collagen-related vascular disease, proximal muscle weakness, and it improves with repetitive effort and more susceptible to SCH and non-depolarizing neuromuscular blockers. Titrate small doses of non-depolarizing muscle blockers to quantitative twitch monitoring, and orthostatic hypotension may be present in these patients. Okay, so finally, Lambert-Eaton syndrome means, remember these 2 points, caused by IgG antibodies, and then it leads to increased sensitivity to both succinylcholine and non-depolarizing muscle relaxants. So finally, just briefly summarize the topic. We have seen the definition and then the structure of the neuromuscular junction, that is presynaptic membrane, synaptic cleft and postsynaptic membrane, and then about the ACH synthesis, storage and release we have seen, and how the ACH molecules present in the synaptic vesicles will fuse and then by exocytosis that is released, and the sequence of events which take place at the neuromuscular junction, and then we have seen the sodium channels and calcium channels, ICH receptors, and finally the clinical neuromuscular blockers, that is one is by preventing the release of ACH and other is by blocking the ACH receptors by competitively, and that is depolarizing and non-depolarizing. And then finally the applied aspects, Myasthenia Gravis and Lambert-Eaton or myasthenic syndrome. Okay, so I hope you understood all these topics, that is entire contents of the neuromuscular junction. So if you are not having books, please try to see the PDF of various books available, that is Guyton, Genong or Best and Taylor, Bunn and Levy, and then read the topic day by day. So you can easily understood when you come back to college, and try to make the notes of all these topics. Okay, so that's all for today. Thank you all.